Good day, this is the Professopher, CBMD PhD, and today's Monday, January 16th, 2023. It is 7:55 and 1 second, a.m. Eastern Standard Time, and today's research, cell identity. So let's do it here. Machine learning method improves cell identity, understanding. Date January 13th, 2023. Source: Carnegie Mellon University. When genes are activated and expressed, they show patterns in cells that are similar in type and function across tissues and organs. Discovering these patterns improves our understanding of cells, which has implications for unveiling disease mechanisms. The advent of spatial transcriptomics technologies has allowed researchers to observe gene expression in their spatial context across entire tissue samples. Although new computational methods are needed to make sense of this data and help identify and understand these gene expression patterns, researchers untangle the role different spatial patterns play in the overall gene expression of cells in complex tissues like the brain. It does so by representing each pattern with spatial metagenes, groups of genes that may be connected to a specific biological process and can display smooth sporadic patterns across tissue. The researchers analyze spatial transcriptomics data from brain regions in mice and humans. They leverage the unique capabilities to uncover the landscape of the brain cell types and spatial patterns. The researchers were inspired by cooking when they chose the name. You can make all sorts of different flavors with the same set of spices. The cells may work in a similar way. They may use a common set of biological processes, but the specific combination they use to give them the unique identity. When applied to brain tissues, researchers identified spatial patterns of cell types in the brain more accurately than other methods. It also uncovered new expression patterns of brain cell types to learn spatial metagenes. The new scientific method has the potential to advance spatial transcriptomics research and contribute to a deeper understanding of both basic biology and disease progression in complex tissues. And this is the Professor, Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.